Hi everyone, I'm Xue Aihuiping, a postdoc from National University of Singapore. In today's talk, I am delighted to share our research on individual-specific cerebellar networks. Our study first explores the detailed organization of the cerebellum using two intensively sampled participants. Then, we expanded our investigation to include a more extensive dataset, allowing us to quantify the individual differences of cerebellar networks. Cerebellum is connected with the cerebral cortex through this closed-loop cerebral-cerebellar circuit. It's hard to study because it's polysynaptic. In animal studies, scientists can use virus tracing techniques. But when it comes to human studies, functional connectivity is a powerful tool to investigate this circuit. Functional connectivity is a quantitative measure used to assess the functional interactions occurring between different brain regions. And resting state functional connectivity has been widely used to characterize large-scale cerebral cortical networks. Cerebral cortex and the cerebellum are functionally connected. In this video, as the seed is moving across the cortical surface, there are observable changes of functional connectivity patterns in the cerebellum. Similarly, when the seed is moving in the cerebellum, the functional connectivity patterns on the cortical surface is also changing. The functional connectivity between the cerebral cortex and the cerebellum enables us to map the cerebellar regions based on the cerebral cortex. Previous work from Bergner used Yale 2011 cortical parcellations to estimate cerebellar parcellations at the group level. According to Bergner's study, there are multiple representations of the cerebral cortex in the cerebellum. The primary map starts from the motor region, follows with the association networks, and ends as a default network. Then a mirrored secondary map begins as a default network and ends at another motor region. The third map is at the farthest extent of the posterior lobe. And now, let's shift our focus to the individual level. In our 2021 paper, we estimated individual-specific cerebellar parcellation on two intensively sampled participants. These two participants each completed 31 sessions, comprising about 60 runs of resting state data. We first used a multi-session hierarchical Bayesian model from our group to obtain individual-specific parcellations on the cerebral cortex. Then we use a winner-take-all algorithm to assign cerebellar voxels to cerebral cortical networks. Because the multiple representations at group level, we are also able to find these representations in individual level. The individual cerebellum follows a similar organization as the group level, but with additional intricate details. Here we use the flat map to visualize the cerebellum. The three representations are also shown on flat maps. The primary map here, then follows the secondary map, and finally the small tertiary map here. The parcellations of the two participants share a lot of common features, but there are also some noticeable differences between them. For example, the default networks in black circles can be found in both participants. But in participant 1, there's a language representation in the green circle, while participant 2 does not have it at the same location. The default A network in the blue circle is also absent in participant 2. To examine these individual differences, we use seed-based analysis, placing seeds at the same MNI coordinates for both participants. And now I'm showing the functional connectivity patterns on the cerebral cortex, with the black lines marking the boundaries of the corresponding cortical networks. We can see that the seed map aligns well with the cortical networks. In participant 2, the functional connectivity maps are very different from participant 1. Therefore, the differences in network topography are the results of distinct functional connectivity patterns rather than our parcellation algorithm. These two intensively sampled participants give us some really detailed results, but collecting so many scans is not easy. And unfortunately, with only 2 participants, the analysis we can do is very limited. Our next project applied the same method to the Human Connectome Project. Although each HCP participant only had 4 resting state runs, the number of the participants enabled us to conduct quantitative analysis. Similarly, we first estimate the cerebral cortical parcellations. Here I'm showing several examples. Then, we use the same winner-take-all algorithm to estimate the cerebellar parcellations. Both cortical and cerebellar parcellations reveals individual differences. With the parcellation completed for all participants, we focused on exploring network lateralization. Let's take the language network as an example, which we know that it's typically left lateralized on the cerebral cortex. The participant has larger language network on the left hemisphere. Conversely, on the cerebellum, it is larger on the right hemisphere, as the cerebellum connects with the contralateral hemisphere of the cerebrum. To measure the lateralization of each network, we used a normalized network size difference. This value is computed for both cerebellum and cerebral cortex, each participant and each network. This normalized network size difference is a value ranges from -1 to 1. Positive value means left lateralized and negative values means right lateralized. So to visualize this, we can draw a scatter plot. The x-axis represents the cerebellar lateralization, and the y-axis represents the cerebral cortex. Here is an example of the language network, and each point is a single participant. And here the histograms show the distributions of the normalized network size difference. The language network is left lateralized on the cerebral cortex for most participants. And in the cerebellum, the language network is right lateralized in most participants. To assess the consistency of network lateralization between the cerebral cortex and the cerebellum, we calculated the correlation between the cerebellar lateralization and the cerebral cortical lateralization. And for this language network, R is -0.75. On the other hand, the visual central network exhibits lower lateralization in both cerebellar and cerebral cortical networks, with a correlation of only -0.1. This indicates less consistency in cerebellar lateralization compared to the language network. And now, take a look at this bar chart showing the correlation values for each network. And higher absolute value of this R means higher consistency. As we can see, the association networks exhibit higher consistency, while the sensory motor networks shows lower consistency. Now, let's explore whether the higher cerebral cerebellar lateralization consistency is association networks is due to greater lateralization or not. We can plot the mean lateralization in the cerebellum against the consistency. The y-axis is the consistency, the same as the left figure. And the x-axis represents the mean lateralization in the cerebellum. We notice that these association networks always show higher lateralization consistency, regardless of the mean lateralization level in the cerebellum. Next, we aim to assess the alignment between our cerebellar parcellation and the task activation patterns. We know that different networks are activated by distinct tasks, such as the control network being activated during working memory tasks and the motor network being activated during motor tasks. For example, the tongue movement specifically activates somatomotor B network. Similarly, the task contrast values are higher within the somatomotor B network in the cerebellum. To compare the activations between networks, we average the positive task contrast values within each network. We computed for all participants and put it in a box plot. The task activation is significantly higher in somatomotor B network. And for another example, we have this working memory task. I'm showing the results of the 2-back versus 0-back contrast. The control networks are more activated in the cerebellum and in the cerebral cortex. For a single participant, the task contrast in the cerebellum is not as clear as the task contrast on the cortex in HCP. But when we look at the whole population, we can still see a clear distinction between the activated networks and the rest in the cerebellum. Then we come to the last question. Is the cerebellar pixelation related to human behavior? To dig into this, we use a kernel ridge regression model. In short, we use a weighted average of the behavior scores from our training participants as a prediction of the behavior score of a test participant. Training participants will weight more if the population is more similar to the test participant. And population similarity is measured by the spatial overlap of cerebellar network between participant pairs. We use 58 behavior scores directly released by HCP, as well as 3 derived factor scores that represent cognition, life dissatisfaction, and emotional recognition. And here's the prediction accuracy using cerebellar network topography. For cognition and life dissatisfaction, the average accuracy is 0.25 and 0.14. We use the same setup for the cerebral cortical network topography, and it shows the same pattern. But the prediction is better for the cerebral cortex. So this could be because the cortical data is of better quality and can capture individual differences better. To conclude, individual differences in network topography exist in both cerebellum and cerebral cortex. Multiple representations of the cerebral cortical networks are found in individual cerebellum. Association networks have higher cerebellar lateralization consistency than sensory motor networks. Individual cerebellar networks conform to task activations. Cerebellar network topography can predict human behaviors. Thanks to our funding sources. Also thanks to my collaborators and lab mates. Our postulation code is available here. Thank you.